Assalamu alaikum. My name is Shoaib Hussain. My ID is 202353112. Today I am going to present my final defense on the thesis deep learning based instant segmentation of leaf diseases. I have done this thesis under the supervision of Nurjaman Faruqi sir. Here are some of the table of contents that I am going to talk about. In this you can see there are introduction, background, research background, motivation for the research, research objective, research gap, research question and many more. We will talk about this in the next slide. First of all, there is introduction. So first we need to define why I am doing this project. The project is deep learning based instant segmentation of leaf diseases. So let's divide this into different chunks. First we need to we have imagine we have disease leaves, different type of diseases on leaves such as corn, common rust or potato early blight. So we will pass this to a deep learning based model. That deep learning based model will be any type of segmentation model. Then that segmentation model will basically not only classify the disease that particular leaf have, but it also classify the pixels. Each pixels belongs to a particular disease. So that is the whole concept of the thesis. So why I am doing this thesis? So main reason I am doing this thesis, for one of the reason, leaf disease poses a significant threat in Bangladesh agriculture sector. There are lots of crop, but main crop in Bangladesh are rice, potato and corn. So I will be focusing on these three crops mainly at the initial stage of the thesis. So one of the reason I am doing this, because I think if I can create a deep learning model which will help to automate the whole process, then the farmers or the other people who are in the industry of agriculture don't need to rely on the manual laboring to find out the leaf disease. Rather it would be an automated process which would be much faster and more accurate, less error prone. That is the one of the main purpose behind the thesis. Now let's talk about the background. So background, first, during the thesis I have studied almost 27 to 28 research papers on the thesis of segmentation of leaf diseases. From that, what I have found that most of the theses are not directly related to segmentation of leaf diseases. There are lots of theses are based on deep learning classification, but segmentation is not that popular, specifically leaf diseases. But I have selected 8 papers which are most relevant to my research. Then I have tried to do some background checking, and background checking was the rural communities where access to expert knowledge and resources is often limited, which I have talked before, that if we want to automate the process, then it can solve these issues where there are not enough resources in the rural communities, and also the manual process of detecting leaf disease are time consuming and costly. And lastly, if we bring that deep learning segmentation, then we can avoid all these issues. Now let's talk about the motivation behind doing this research. As I have told before, Bangladesh is an agriculture country, so its economy depends on agriculture, but there are huge problem of crop diseases. So if there is many crop diseases in Bangladesh, then the economy will fall. So there is need to a step to solve this, not only solve this but automate the whole process. That's why I am trying to do a thesis based on detecting the most common crops in Bangladesh. Those could be rice, potato and corn. These are the most common. And then the current challenge is create a data set. Not only the data set but label data set with proper annotation. Here I am talking about annotation of the marks. We not only want the image to have a label, but we also want the image to have a mark, so that we can train a segmentation model. And the last thing is, as I have mentioned just before it, annotation, specifically marks annotation. It is probably the most difficult and time consuming uh, and tedious work in deep learning. It requires lots of time, it requires lots of patience, and it requires lots of people at the same time to annotate these images one by one with each particular disease. That's why I have proposed a new methodology, which would be semi-automated annotation pipeline. In semi-automated annotation pipeline, we don't need to do the manual annotation from the scratch, but it would be done by using a deep learning model, and then on top of that model we'll use an annotator, human annotator, to correct the prediction. I will talk about this in details in future slides. So let's move on to the next slide. Here we have the objective and possible outcomes of the thesis. The first of all, the objective is a segmentation model, instant segmentation model for crop, for potato, corn and rice leaf diseases. There would be total 10 type of diseases, where potato have 3 diseases, corn have 4 diseases, and rice have 4 diseases. So total would be 11. After that, we would be create a semi-automated annotation pipeline, which will help us to annotate data, which we can increase the size of the data, and the more data is better for deep learning. The final outcome would be a new annotated data set for rice, corn and potato leaf diseases, an effective semi-automated pipeline for data annotation, a real-world application based on real-time inference. So basically, if we deploy the model, then we can have a real-time inference website or web app. But that's outside scope of the thesis, but we can achieve it using that particular segmentation model. Now next, let's move on to the next slide. Based on my reading the research paper, I found some gaps in the previous research. The gaps were, most of the research field only focus on single crop type, and there are data diversity, issues of data diversity. And the second problem was data is scarce for this task. So leaf disease don't have many data set available, and not only that, if there are image data, then there is not enough annotation for that. After that, we have most solution tends to avoid deep learning. So deep learning is a time consuming task, because it requires lots of time to training. Not only that, it can be computationally expensive. That's why people try to avoid it. But recently deep learning is improving faster. At the same time, the competition also getting better and better. So deep learning can be a better solution, specifically if we are trying to reduce the error. Now let's talk about the research question, like what would be our objective, what could be the challenges we could face. So first of all, the data set diversity, like do we have enough data set, do we have enough diversity in our data set. Second, the problem is annotated data set. Not only having data set is not enough, but we need to have the annotation for each of the images, for each of the crop disease to be marked. Computation cost for training and inference. Since this is an instant based deep learning segmentation, then we need to have GPUs for training, and GPUs can be costly. So this is also a big concern. Developing a robust model. A robust model is a model which not only perform in a testing environment, but it also can be performed in a real life practical scenario. So this is all the challenges that we could face. Then we talk about literature review. In a literature review, I have included total 8 papers, where I have discussed some of the methodology and the limitation of those papers. So first we have Mohammed Shoaib. The research was done in 2022. The methodology was quite interesting, I would say. They use DCGAN network to create new generated images. Then they use contextual aware net to segment those images, and then after segmenting the crop disease, then they pass those segmented part to a 3D CNN. The first problem is class imbalance. There in the training data there was lots of class imbalance because most of the images were having only good category of crop. There were not many bad category of crop. This is one of the thing, and the computationally is very expensive. First of all, we can see there is two different model, one for segmentation and one for classification. For segmentation they are using CANNet. For classification they are using 3D CNN. And also DC GAN is really computational expensive. It's a variant of generative adversarial neural network, and it requires lots of competition to generate an image, so it is not quite practical. Second, we also have another research from Mohammed Shoaib. It also published in 2022. Here he uses UNet for the segmentation and inception V2 and inception V3 net for the classification. But the problem is the image is unable to segment disease area, and also the two different neural networks for segmentation and classification, which can be computationally expensive. Second, we have Vijay Singh. Vijay Singh did not use any deep learning methodology, but it was interesting because he uses genetic algorithm for the segmentation part and the SVM, the support vector machine, for the classification part. But the problem is there is no deep learning, so the accuracy would be not as good as any deep learning model. I am assuming that, I'm not sure about that, but I am assuming that the accuracy might be lower than that. And also the dataset size was very limited, and dataset was not open source also. Then I have K. Elan Gopan. He uses K-means clustering for the segmentation and SVM for the classification, also similar to Vijay Singh. And also the limitation was traditional method for segmentation and only limited to chili leaf. Then I have Parul Sharma, who used two different network. At one is SCNN and another is FCNN. The single it is limited to single crop and unable to segment the disease area. Then I have MD Ershadul Haq. He used manual data annotation, then he augment those data, and he used YOLO V5, one of the variants of YOLO, to detect the images. So it is not a segmentation problem, it is a rather object detection problem, and he did it for only for rice crop. Then I have Fatima Zara. Fatima Zara used deep CNN, SGD and ADA, but this paper did not have any any segmentation related work. It was mainly because of the classification solution, and the model, there was no model diversity. Now let's talk about my methodology, what I am going to use. For me, first of all, I am going to collect the data. Then after collecting the data, I am going to clean the data. After cleaning the data, I am going to split the data into train and validation set, and the reason is doing that early because I want to select the validation set by myself, so that the validation set and the train set are similar, so that the model performance can be justified. After that, I have to do the manual data annotation. In the initial phase, I have to do the manual data annotation for some images. Then we move on to the semi-automated annotation pipeline. I will be discussing about this in details in the next slide, but the whole purpose of semi-automated annotation pipeline is to create use deep learning model to predict new marks, and human annotator will fix those. Then we have a dataset from a semi-automated annotation pipeline. Then we combine those dataset with manual data annotation and the semi-automation. After we get two dataset, data augmentation. We do data augmentation, then we train a model, three model EULO V9C, EULO V8L, EULO V8M. Right now, three of the model are the latest model from EULO family. We train that model, but for two different variants of dataset. First of all, we tried actually three variants of dataset. I actually tried train the manual data annotation only. In the three different dataset, I tried two data augmentation, and I also tried the combined datasets only without any augmentation. I will be talking about those in details in the results section. Then we use evaluation mean average precision for it, one of the classical evaluation metrics for segmentation tracks, PR curve, precision recall curve, and confusion matrix. Now let's talk about semi-automated annotation pipeline. The whole purpose of semi-automated annotation pipeline is to reduce the annotation time, and also uh, try to uh, generate more images, annotated images. Okay, now let's see how this works. First of all, we have a fine-tuned EULO based on manual annotated data set. So basically, we have initial step, we have a manual data set. Then manual data set will be go into the EULO V8 small model. The reason I am choosing EULO V8S is because it is smaller and it can be trained really fast. So after fine tuning it, we will choose new unseen image, the images that was not in the data set. It is some new images. We will then split those images, train and validation split, and then we will pass it to the EULO V8 as to inference those images. EULO V8, which is trained on the manual annotation, will inference on those images, and that I have found a problem. The problem was the EULO V8S was generating too much point for a simple polygon but simple marks. That can be really problematic, because if I want to edit those, then editing those marks can take too much time, which is not efficient at all. That's why I use an algorithm called RDP. So in that algorithm, what it does is it simplify the polygon, so it reduce all the marks, and it reduce all the points in the marks without changing the marks shape. After simplifying the polygon, I save the predicted marks as a JSON file, which I can load back in my local machine using my annotation tool, which is VGG annotation tool. After that, I can manually recorrect the marks. Here I don't need to recorrect, I don't need to marks them from the scratch. I can do that manually. I can just recorrect those marks that is provided. That could really change, that could really reduce my time for annotation. Here is the code for my pipeline. So first of all, I am like getting all the images, and I'm loading the model that is fine-tuned, and then I'm simplifying the polygon, and after that I'm predicting using my model function. Then I'm getting the info, and this is required for my annotation tool. So after that I'm creating a JSON template. That JSON template will have all the information that could be required to load those images in annotation format. And finally, we have the automate annotation function, which will automate all the tasks. So all I need to have to do is pass the images and the model. It will automate all the images with the annotation as a JSON file, and I have two different. After that, the finishing product will look like this. Here I have this. This is predicted by the model, that the front gray leaf, so if I want to change, like suppose I don't like this, so then I can like basically edit those. Here, edit those. So you see, I don't really need to edit from scratch. All the other like 13 marks were edited before. Right now all I'm focusing on the recorrection. This does reduce time a lot. After that, we can focus on the result. First of all, I am focusing on the mean average precision for bounding box. So there are two type of values. One is mean average precision for 0.5, and another one is mean average precision for 0.5 to 0.95. I won't go into the details, but the first thing that I've noticed that the YOLO V8M that is trained with manual annotated dataset and the YOLO V8M that was trained with a combined dataset, we can see an increase. Like the YOLO V8M was performing 0.7389 at at MAP 0.5, where YOLO V8M with combined dataset were performing 0.754. So we can see an increase. So we can conclude that increasing the dataset size does it affect the model, and we can also conclude that semi-automation pipeline is helping us to increase the dataset size. And the second, we have the segmentation marks. We can see the same thing, like manual annotated dataset for YOLO V8M is scoring 0.7 to 78, where combined dataset is scoring 7347. It's a minor improvement, but it's still an improvement. So increasing the dataset size does matter. But in this chart, we can do see that YOLO V8L with combined dataset perform the best. So now let's talk about what is my contribution. So first of all, I did build a disease dataset. I have taken this dataset from two different sources. One is from Plant Village and another one is from Dhan Samadhan. So I have annotated almost 3900 images by myself, then manually annotated. Then I introduced a new technique, semi-automated annotation pipeline, where I annotated 800 images in 3 days. And my observation, comparing my manual annotation time and the semi-automated annotation time, was that the semi-automated annotation time was reduced to 85%. That was huge. Now the conclusion is I will create a new data set. In the limitation part, the images that are collected from secondary sources such as Plant Village and Dhan Samadhan are mainly indoor images. They don't have any background in the video, and this could be a problem for a real life scenario where it would be used as in the field and the field will have a background. The semi-automated annotation pipeline only works with one type of annotation tool, which is VGG image annotation tool, and data set contains only 10 different crop diseases. I intend to increase the size in future. So future work, we can, from the limitation we can derive the future work. The future work requires we need to manually collect outdoor images for disease crop. We need to expand the uses of semi-automated annotation pipeline for more image annotation tool, and finally we need to expand the class number using the semi-automated annotation tool, so that we are not limited to 10 class, we can go further. So that was all for my presentation for my thesis final defense. I hope you like it. Thank you very much.